Hello viewers, welcome to Amir Hamza, Biotech for you. Welcome back friends, I hope you all well. My previous video, PCR-based cloning primer design, I showed you how to design a primer in the molecular cloning part 1 primer design. Today I will show you how will you conduct your PCR reaction. Everything I will explain. How will you set your annealing temperature? How will you set cycle? And that's all. And this part is molecular cloning part 2 PCR. So after this video, I hope you all will understand how to conduct your PCR after designing your primer. And I also help you to calculate the temperature and the basic requirements for a primer for the cloning. So guys, let's get started. For complete understanding, keep watching until end without skipping. Step 1. Optimize the primer and set the annealing temperature. So according to this optimization of your primer, as you remember, I designed the primer of Homo sapiens cathepsinel escape variant 2. So this primer you have to optimize and set the annealing temperature. Copy this forward primer and open the browser. I already set some website link. I will give you all in the description. Just paste here and calculate, and you can see the length of your primer 32 and the melting temperature is 66 degrees centigrade and the GC content is 53, and as you know 40 to 60% of GC content is required for primer. So I think my forward primer is passed this optimization, and second step is this same primer we will check the cell annealing, Ctrl V, and calculate. Here also same thing, but I think two different website it provide two different kind of temperature. They showed 66 and here is 65.7 more precisely. So I will click the check self complementary. So as you can see here, the potential hairpin formation, so we cannot use this primer for the PCR because we consider the first and second none, but third there is any problem in like this red color, then no problem. But this one complementary is none, but potential hairpin structure is as you can see there are 3 replica, and the hairpin structure from the I provide restriction site and mother sequence. So I could do the change restriction site. So here I use the GGATCC is the Parmesan site. If I change this into eco R1 site, that means GAAETC. Now just copy and paste here 63.1, no problem. So as you can see, the hairpin formation is now only one, and between them okay. Now if I delete one bases here and then click, so now primer is okay. You can see potential hairpin formation is none and complementary is none. So we can use this primer for your PCR reaction. So the temperature we found 66 degree centigrade. Now do the same with the reverse primer. So I am doing the same. So I will go fast forward. In this case you can see GC content is 66%, so it is so high. So we can change some. I just delete one basis from the end and I will add here AA or TT. Okay, now 50%. Now we will check the complementary, and you can see 67 degrees centigrade is the melting temperature. So paste here and check the self complement. So this already perfect, both are none, so we can use this. So our forward primer and reverse primer is ready by optimization. Now we can write here 67 degree, 67 degree. And remember one thing, this forward primer and reverse primer, temperature between these two primer, this one is 66 and this one is 67. So remember, it should not cross the more than 5 degree. Okay, so here our Temperature difference between forward and reverse is only 1 degree centigrade. So our optimization is finished, and the melting temperature would be forward primer plus reverse primer divided by 2, means the average value. Here we can find 66.5 degree centigrade is the annealing temperature. Okay. Step 2, PCR reaction and select polymerase enzyme. Set the time Tm, that means temperature, and cycle. Okay, go for it. Now, polymerase chain reaction. The basic information of polymerase chain reaction is technique for generating large quantities of a specified DNA. That means that DNA you want to clone, or that DNA you want to identify, or that DNA you want to make multiple copies for your experiment or your research. Second, PCR is a cell-free amplification technique for synthesizing multiple identical copies of any DNA of interest. Principle of PCR. Number 1. The double-stranded DNA of interest is denatured to separate into 2 individual strands. Second, each strand is allowed to hybridize with a primer, that means renaturation. Third, the primer template duplex is used for DNA synthesis, that means the DNA polymerase. This DNA polymerase actually helps to synthesize new DNA. Fourth, denaturation, renaturation and synthesis are repeated again and again to generate multiple forms of target DNA. So fourth step is actually talking about the cycle. How many cycles you run for your PCR, and depending on this cycle you will get a number of DNA, that means the millions copy of your desired product. Okay, now I will talk about the PCR cycle. Okay, this is your target DNA, and this DNA you want to make amplify by so many cycles. So here is the cycle 1, first denaturation at 95 degrees centigrade, and this time polymerase comes out and also primers that you mix in your PCR tube and free nucleotides, and then at the annealing temperature this binds to the specific region of how you design, and then extension at 72 degrees centigrade. And the free nucleotides are bind, and then it makes the replica of this DNA. And then again cycle 2, denaturation, then annealing, then again extension. And continuously this work, how many cycles you run, it will be continued. So in case of molecular cloning, we use 34 cycles for desired product and multiple copy of our desired DNA. In this section you can see as the DNA segment is become shorter, and this would be your target DNA after PCR product. You can see the mention the target DNA, and these 2 copies of DNA in presence of so many cycles in time, it will be makes millions of copy. And this is all about the cycle, and the concentration of your target DNA is increased by the run cycle by PCR. So millions of copy are ready by PCR.